Yep. Hello, everyone. Thank you for joining us. A warm welcome to those of you who are watching through the live stream. This is IBD&Tea, an online event that we've organized so you can ask the experts working at the forefront of inflammatory bowel disease, both treatment and diagnosis. So I will be hosting this afternoon. My name is Bruno Martin. I work at the science communication company Science Seed in Spain, and we are part of New Deal, which is a research project funded by the European Commission centered on the development of a novel treatment for inflammatory bowel disease. Now that everyone is at home and we're all locked up, we thought this is probably the perfect chance to organize an informal event where we can chat with the experts directly. So for this event, New Deal has partnered with Euphoriah. Euphoriah is another European project funded by the European Commission, centered on IBD research and particularly on monitoring and diagnostics of inflammatory bowel disease. So let me do some introductions. First of all, we are happy to be joined by Rachel Sawyer from the UK. Rachel is an IBD patient and she shares lots of interesting information about the disease on her Twitter account, The Bottom Line. So you can go find her at the bottom line, @bottomline_IBD. Thank you for joining us, Rachel.
Thank you, Bruno, and thanks for the invitation.
We're also joined by two experts who will answer our questions today. So on behalf of the New Deal project, we have Dr. Azucena Salas. She is the principal investigator at the IBD unit in IDIBAPS, which is a research center in Barcelona. Welcome, Azucena.
Thank you. Pleasure to be here.
Thank you. And from the Euphoriah project, we have Dr. Max Waldner. He is professor at the Gastroenterology department of Erlangen University Hospital in Germany. Welcome, Max.
Thank you also from my side for being here and the introduction.
Thank you. So we're going to hear from all of our experts in just a minute. But first of all, if you are watching us live, I'd like you please to submit your questions. So we are here to ask the experts. Please let us know via the chat on Periscope or via Twitter what you would like to know about inflammatory bowel disease, specifically with a focus on treatment and diagnosis. So for the next hour, I will be asking your questions to our experts. To complete the introductions, I'd like you to know a bit more about yourself. So Rachel, can you tell us a bit about your background, just very briefly, and what your role is going to be in this live debate?
Yep, certainly. So I'm Rachel Sawyer. I'm an IBD patient advocate running Bottom Line. Thank you for the kind introduction there. And also I run IBD Women on Twitter, which is a Twitter account specifically for women's matters in inflammatory bowel disease.
Thank you, Rachel. So Azucena, can you tell us a bit about what you will be talking about today?
Yes, so I'm Azucena, and I'm a researcher and investigator at the IBD unit in Barcelona, Hospital, Clínic, and IDIBAPS. And I'm here to talk about, the research that is being conducted currently to develop new therapies or to optimize the current ones.
Okay. How about you, Max? Can you tell us a bit about your angle, your background?
So I'm a physician on one side, and we are having a very large outpatient clinic here on IBD patients in Erlangen. On the other side, I'm also a researcher, and my main focus here is new imaging technologies, mainly also for usage in IBD patients, where we can evaluate disease activity and monitor treatment.
Okay, thank you. So remember, everyone, if you're watching live, you can submit your questions now that you know what everyone is talking about. I'd like to get a bit of a more personal background this time. So first of all, because we're going to talk about IBD, and obviously you all have very different backgrounds, I want to know how IBD came into your lives and how you have come to be connected to it. So in your case, Rachel, you are a patient. Let us know a bit about your story. When were you diagnosed with IBD? How did you decide to become an advocate?
Well, I was diagnosed 20 years ago when I was 31 with ulcerative colitis, actually, first of all. Then I developed a perianal fistula. So it was reclassified as Crohn's. That was 8 years afterwards. And about 5 years ago, I decided that I wanted to know more information and I wanted to share more information. And I felt that the patient education side of things could do with some boosting. So I decided to set up the Twitter account, and really it's gone from there. So along with what is an international collaboration of brilliant IBD advocates, we are really trying to raise the bar for patient care and also to provide a conduit really really trying to raise between the medical community in IBD and the information which is accessible to patients, with the aim ultimately of improving quality of life and information for patients with IBD.
Okay, thank you. What about you, Azucena? How did IBD come into your life? How have you come to start researching on this subject and develop your career with it?
So yes, I like I said before, I'm a scientist, so I'm a basic scientist. I do full-time research, and I started, over 25 years ago, working on research. And since the beginning, I've been focusing on inflammation. My whole career has been on understanding how inflammation occurs. And 20 years out of those 25 have been devoted to IBD, because I worked at the IBD unit at the Department of Gastroenterology here at Hospital Clínic. And they are, for the last 20 years, so we've been developing and increasing this research unit. So our research focus is to understand really what's going on in the patient, in the tissue, when the disease develops, when there's a flare, when the inflammation is going on. And, so really what's going on, it means looking into the cells, looking into the proteins, looking at what genes are turned on or off. And the objective, the final objective, obviously, is to understand these pathways that are important to lead to disease, so we can target them, so we can treat specific molecules that are involved in this process. I know, a lot of the patients, as they are very familiar with the treatments that are available today, and they will know that 20 years ago, there's really the only treatment for IBD was corticosteroids or immune suppressants, nothing specific. But since anti-TNF or infliximab was introduced, that has changed. So there are now more options. We have more monoclonal antibodies towards other molecules, not just TNF-alpha, but and even small molecules that can be taken orally, recently developed and approved for UC, for ulcerative colitis. But all these new drugs have come out because of the research that was developed around the globe, really, to understand the pathophysiology, these pathways that I was talking about. So without that knowledge, we cannot lead to new targets, to new diseases. So our lab really focuses on that, and that's where my interest is. And I think this is a fascinating disease, a very complicated disease, and from the point of view of the immune system and understanding the biology of it, it is a really interesting scenario. But our final goal is to really improve the patients. And just a sentence that I would also like to say is that one of the important areas of research, now that we have many therapeutic options, is to understand why some patients fail drugs, why there's so many patients that do very well, for example, with infliximab, but then they fail, others that fail completely to respond, or they just lose response over time. And now that we have other options, the more we understand how these treatments work, the more we can also adjust them to each patient. The goal, which is still a dream, I think there's really nothing that we can use today in the clinics yet, but the idea is to establish a personalized treatment so that you can really choose patients based on their biology with the best and partner them with the best drug.
That's really interesting. Thank you, Azucena. We will come back to this point of personalizing treatment. I think a lot of people watching us will be very interested to know that. But first, let's hear a bit from Max as well. So Azucena has just told us that she's a basic researcher. She's trying to make the patients' lives better from the very first steps where you come up with new drugs. You, Max, I understand, are also a clinician, so you are seeing patients in your day-to-day from a closer perspective. Tell us a bit about your background and how you have come to be involved with IBD patients and research.
So I actually came to IBD research via our boss here in Erlangen, is Markus Neurath. I met him in Mainz, where I started my medical career after finishing university. And he already there was completely devoted to research and also clinical therapy of IBD patients. And somehow he motivated me very strongly to go also in this research direction. And then we moved to Erlangen 10 years ago, something like this. And Here we have a very large outpatient clinic, as I already said, and also a lot of research going on. My personal interest is a lot to imaging technologies and to kind of pursuing new things, how they could help patients. And also in this direction, it's very interesting that we have new photonic techniques that are currently not in the clinics, but they can go to the clinics and help here also for maybe individualized therapy of patients, especially also in IBD.
Thank you. So I'd like to just start off from there because I have some questions. You just said your team works on new like optic and acoustic methods to monitor IBD. Can you tell us a bit about how they work and how they compare to how IBD maybe is usually diagnosed?
So usually when you have patients here in the clinics, you have an initial diagnosis of the type of disease, ulcerative colitis or Crohn's disease. And to make the diagnosis, you usually need different kind of components regarding technology. So you need, for instance, the clinical picture, you need maybe some information from conventional imaging techniques like ultrasound or even tomographic techniques like MRI. The most important aspect is actually endoscopy. So you have kind of characteristic changes of the mucosal surface in Crohn's disease and ulcerative colitis. And then you additionally, by taking biopsies, get a microscopic evaluation of the tissue where you also have some specific changes like granular formation in Crohn's disease, for instance. And by putting all these things together, then you can make the definite diagnosis of either Crohn's or ulcerative colitis. So this is one part. The other part is always monitoring disease activity, and especially also when you start a new treatment that you maybe want to see very quickly how the patient really responds to the treatment, if he is responding or not. And there on one side, you can also do endoscopy, but of course you wouldn't want to do this every few months. So most patients, they are quite unhappy if you already after half a year tell them that you would do another colonoscopy. So therefore we need other methods that we could use. And one very frequently used technique in Germany is ultrasound, because you can already get a lot of information with ultrasound. You can look at the thickness of the intestinal wall and also perfusion with Doppler ultrasound. However, you have not so much sensitivity. So if the patient has just maybe very slight differences of activity, you wouldn't see it with conventional ultrasound. And that's where other techniques come in. We found one technique several years ago, that's optoacoustic imaging. That's a technique where you actually use light to kind of excite certain molecules in the tissue. And these molecules are molecules that have a very specific absorption characterization. So like hemoglobin, for instance. And then if this light is being absorbed, the molecules develop vibrations. These vibrations you can then detect by ultrasound. And the advantage is that by using light for excitation, you can do molecularly specific excitation. So you can really just excite hemoglobin, for instance, especially if you use several wavelengths sequentially. And then by detecting the light with ultrasound, you have a very high penetration depth. So when you usually have light-based techniques, you can maybe just go into the tissue for maybe 100, 200 micrometers. If you just use light for excitation, it will go into the tissue for several centimeters. And the ultrasound signal that gets out is also from several centimeters into depth. So what we somehow realized is that if we use optoacoustics, we can detect hemoglobin levels, so the perfusion in the intestinal wall through the abdominal skin. So just like in conventional ultrasound, so you go to the doctor, you're kind of lying on a bed, and then we are doing an investigation or a procedure that's taking just a few minutes. It's like ultrasound, and you can quantify the blood content in the intestinal wall. And the blood content is kind of a surrogate parameter of inflammation, because we know kind of for maybe even more than 100 years that in acute inflammation you have kind of increased perfusion. You have, if you have a inflammation in the skin, you can see redness, what is corresponding to this increased perfusion. You have swelling and things like these. So these are really very well-known signs of acute inflammation. And we found that if you correlate this hemoglobin content in the wall with, for instance, endoscopic disease activity, then you get a very good correlation, and this non-invasively. So you don't need to do endoscopy. Just by doing this non-invasive procedure, you have a more kind of objective information about the real disease activity in the patient.
So you have the advantage of ultrasound, which is a non-invasive examination,
Exactly.
but you have more detail as well.
Exactly.
More
and
information
you have molecular information. So it's not just structural information. So what you get with ultrasound is more kind of really tissue morphology. Here we really get information about single molecules.
Right. Rachel, I imagine this is exciting news for patients, because within the IBD community, you must speak to lots of people who are probably very concerned about the constant monitoring, the checkups, the dreaded colonoscopies. That must be quite a burden on IBD patients to think about these constant checkups. So what do you think about these techniques and how patients might respond to them?
Yeah, well, for sure, as Max was saying, I mean, colonoscopies are really the last thing that anybody wants to have done, albeit they are a necessary evil, in inverted commas. So anything which could minimize the invasive procedure is obviously to be welcome. I mean, obviously, the natural question is, if you find polyps, presumably you still need to go in. So, but as a answer to the question, do patients want to have less invasive procedures? Absolutely, yes. And less frequent visits to the hospital, particularly at the moment. Any system or tests which can minimize colonoscopies is very welcome for patients. Yeah.
Would you say this is one of patients' perhaps recurring concerns, that constant monitoring, or is it something that you just forget about and it becomes part of their routine? Are people on top of new techniques and they want to know what's been developed in this area, or is it more about treatments?
I think I don't think constant monitoring is a problem per se. And I think perhaps after you've been a patient for a long time, it gets hard baked into you anyway. So I think perhaps if you're a newer patient, that may be more of a problem. But as you go on, I don't think it's so much about the monitoring per se, but what that monitoring involves. For example, if you could do fecal sending off samples in the post. So it's not really the test, it's what the test involves, I would say.
Right. Azucena, I'd like to go back to you as well. You were talking about all the current treatments, and treatments are in the pipeline for the future of IBD. I would like to know, just to get a very quick overview for people who might be new to the subject, or like me, who just don't know much about what are the current options. How would you say the treatment prospects for an IBD patient now are different from what they were, say, 15 years ago?
Well, I mean, they're better and worse, I would say. I mean, I think that, 15 years ago, all we had was anti-TNF, which really revolutionized treatment of patients because it's a very potent drug. And if you look at the history of the development, there's really a big gap in between infliximab and all the new drugs that just appeared a few years ago. And that gap was because, despite the many efforts, and if you look into the literature, there's so many trials done in IBD patients that fail, so many drugs that go into phase 1, phase 2, and then either don't pass phase 2, or they never make it, or they make it to phase 3 and then they're discontinued. And that's because we realize, we always compare ourselves to other autoimmune diseases or other chronic inflammatory diseases. And you can see how many of these pipelines are going side by side. But then diseases like, psoriasis, of course, but even rheumatoid arthritis or others seem to respond better to all the new drugs. And then there will also always be, like, UC, ulcerative colitis or Crohn's disease lagging behind in getting new drugs. And that happened for a long time. And in those years, we learned a lot precisely about the failure to anti-TNF, because after the initial boom of anti-TNF, many patients were responding and everyone was very excited. But then we realized with the use of anti-TNF over years that many patients do not respond and many others lose response over time. So the disease may change, the disease mechanisms are changing based on what we see. And so the need for new drugs was obvious. Then, because of all the research done in all other fields too, not just in IBD, but together with other fields, autoimmune diseases or inflammatory diseases, we finally got, the next drug approved, which was ustekinumab, another antibody targeting a protein that is involved in the cascade of inflammation, and that was, hopeful. And after that, two more drugs have been approved. So right now, there's the rhythm of trials. I think it's increasing, although it was already very high. What we have to be is better about choosing the targets. I think sometimes still today, when pharmaceutical companies decide to go forward with a drug, it's not necessarily based on robust evidence, specifically in IBD. Many times it's based on more broad research in other areas, or drugs like, for example, tofacitinib that was just approved, the oral compound that inhibits JAK kinases. Tofacitinib was first approved in other diseases, so is other of the drugs that are being tested now, like risankizumab. Also, they pass, the safety issues. They show efficacy in diseases like psoriasis or arthritis, and then pharmaceutical companies go into Crohn's disease or go into UC.
So drugs that have been proven safe for other treatments that have been repurposed and trying to see whether they are effective for IBD, but there is a lack of
there were many
focus,
failures. There are. I mean, and if you look, for example, vedolizumab is another antibody that has been, I think it was approved in 2014 by the FDA, and it's in use in Europe for several years. And vedolizumab is specific for IBD. It's the only drug we have that really, because it targets a molecule that helps cells, immune cells, make it into the gut, and only into the gut. And so it doesn't make any sense to give it to a patient that has psoriasis or arthritis, because it's not gonna, stop the flux of inflammation in the gut. So that one drug was, developed, the research behind it goes back to 20, 30 years ago, a lot of information on models, on primates, on other species, etc., until this antibody was developed. So that's the only example where we have a drug that has, but I think it's because of all the failures and of drugs that were thought, this is gonna work in IBD, and then the clinical trial will fail and another fail, that companies now are taking another approach. So the next approach was, let's repurpose. Now I think trials are more difficult to succeed because you already have drugs that work to a certain degree. Let's not forget that most of them don't treat all patients. And then more and more companies, these have to be very big companies that can afford these kind of trials, are just targeting patients. For example, they're only recruiting patients that are failures to anti-TNF, for example, which we know are very difficult to treat. And they do this because they know that for them to have a market, they have to be better than anti-TNF, or they have to offer a solution to patients that fail anti-TNF. And so that's a brave approach, but it's also a smart approach, because in most countries, regulation says you have to be a failure to anti-TNF. I don't know how it's in Germany or in the UK, but I know here in Spain and in the US, you have to fail first several anti-TNFs before you go into vedolizumab, for example, if you have UC or.
But Azucena, you were also saying, and Max and Rachel, feel free to chip in here, but you were also saying that it's common for treatments to stop being effective for certain patients after a certain time. So I imagine this is a recurrent problem in IBD, that even though anti-TNF or any effective treatment is working for a while, it's a reality that patients must know that eventually there may come a point where you stop being responsive to that treatment, and then you will want to know about these other options being developed. I'll open the floor to whoever wants to pick up on that.
And I would absolutely say that's so. I mean, as a fortunately for me, I've been well for a long time on one drug, but if antibodies develop, then I feel reassured at the moment that the drug pipeline is quite plentiful. So, but it's a problem for patients. It's a worry that we have, is that we will get to the stage where it's really last chance saloon sometimes. So yes, it's really reassuring for us to know that there are other options.
Are we at the point now where treatments are already personalized for each patient depending on their profile, or are we not there yet with IBD?
I mean, Max, maybe you go first, that you have firsthand, but my answer is no.
Yeah, I completely agree with you. The problem is that we don't know the right markers. So we would like to have them, and we would like to really be able to evaluate in advance before we start which is the optimal treatment for a patient. But we just don't know what are the markers that the patient must have to choose either this or that therapy. So this is something that is urgently be also needed, and also there's a lot of research going on in this direction, but so far not successfully.
No, I mean, it's been I don't know how many years, but for years now, every time you go to a meeting, with clinicians or scientists, everyone talks about personalized medicine. That's the goal. But in my opinion, I completely agree with Max, we don't have any, even though there are papers out there, articles that have tried to identify some markers. Nothing has been really validated, and nothing has really been, which is important. The important test is in the clinics now, so it's being used. That means that nothing is too good, as good that we can use it. And that's one of them. But my personal opinion, in this, is that we're trying to predict before we know what we have to predict. So we're trying to, just take patients, and I've done this too, I'm guilty too, but you take patients at, before you start a treatment, you look at everything you can look at, and then you say, because you treat them, are they responders or non-responders? And then you compare at baseline. But we're really looking at everything we can look at without really knowing what we should be looking at. And I think that's the mistake. We need to first understand how the drugs work, which seems like a very obvious point, but we still don't. Even if I know a patient receives anti-TNF, and this is probably the drug that's been most studied, do we really understand what are the target cells, what are the target, we know it blocks TNF, but what is downstream of that? How is that affecting the biology in the tissue? Why, because if we did understand that, we did know that, then we could try to predict what, if I know what should happen, I can try to predict, anticipate that. But if I don't know, all I know is I'm blocking TNF alpha, and there's a result. Some patients get well, some patients don't. So I think we need to be a little, go back to the drawing board and start thinking about this, not from let's look at everything we can, which is what we have been doing in the lab, and clinicians have been also doing in studies, looking at all them, clinical characteristics of patients before, but my opinion is that those patients are not that different when you have them in the clinic, for what I've heard clinicians talk about. You obviously give them anti-TNF because you think it's going to work for them, but right now there's no way to predict. And now we have not just anti-TNF, but all the other drugs, at least 4 drugs, depending on the disease. And the ideal world would be, let's start them at the beginning with the right, so you don't waste all these years of going through relapses or going through.
And start with a personalized treatment from the beginning, if possible.
That would be the idea. That would be the idea. Even for example, there's studies in children that develop IBD, and looking at a diagnosis and how these evolve over time. And it seems like you could start all patients with anti-TNF, which some people have proposed over the years, an aggressive approach. And then, they see in these studies that if you do that, the risk of developing, like, for example, fistula, like Rachel was talking about, in her case, or fibrosis, it goes down. But then the question is, can you just treat anyone, especially children, with a drug that is a potent immune suppressant? So that's not the ideal. What you want to know is which of these children need to get started anti-TNF, and they have to, and which don't. The same with adults. if you can give them, if you can have these four drugs to choose from, once the patient has a more aggressive disease and doesn't respond to basic, common drugs, which one would be the ideal? Because they have what we call very different mechanisms of action, meaning that they do the job very differently. At the end, you heal the mucosa, but how you go about it to do it is very different. So there has to be something that each one is doing that may benefit one person better than another.
Is that because the mechanisms for the disease acting in different patients are also different? And do they change in time as well?
Exactly. Exactly. So that's one of the things I didn't say earlier, but that's what makes the clinics, but also the lab work very difficult, is the huge heterogeneity of these patients, and I'm sure, Max can say how different, even Crohn's disease patients, may have very different presentations, very different severities, very different complications. The same with UC. UC is more homogeneous, I think. And Max, you can...
So even though the name describes a common set of symptoms, the biological reasons for those may be different in different patients.
Very different. And so you cannot expect all to respond to the same, and that makes sense.
And I want to remind everyone who's watching us live, they can submit their questions at this point, or any throughout in the chat on Periscope or through Twitter. I also wanted to know, you have focused on the safety and the efficacy of new treatments in the pipeline, but Rachel, perhaps you can comment as well as to the priorities that patients have, because as I understand it, when there are new options for patients that come out, one of the things that is presumably very attractive is not just safety and efficacy, but also how convenient they are, whether it's something you can do at home, whether it's less invasive. How many new options and how much awareness of them is there among the community of IBD patients? And what would be sort of interesting from that point of view of something that's currently a bit of a bore, it's a bit of a pain in the neck, and you'd think, well, if treatments could progress towards something that's more comfortable to the patient, that would be a very significant advantage as well. Rachel, hello.
Hi. So I'm not
sorry.
very well at all. So can you hear me okay?
I can hear you well now, yes.
Okay. So
now, the question was, if patients are focusing as well on patients who stay on top of new developments, they're presumably very focused on the convenience of treatment as well, not just the safety and efficacy, but whether it's something you can do at home or something that takes out the invasive nature of some of the interventions. So what are the new options that you know about in this regard and that patients could be more interested in?
So what do I know about that patients may be interested in?
Exactly, yes. Yeah,
so is that, sorry, I'm really struggling to hear you. So I'm interpreting what I think your question is, which is what do I think the patients will be interested in terms of new treatments and other...
Exactly, and specifically in convenience, yes.
Yes. Well, I think two things really. I mean, the one that lots of patients are interested in is home testing for fecal calpro. I have some concerns with that around hypervigilance in patients and whether that's suitable. If people are anxious anyway, is it the best thing to be able to have people testing at home without necessarily the filter of receiving that information after it's been examined by the doctor? But that's certainly something that is popular with patients, the idea of just being able to test their calprotectin at home. And the other one, of course, is subcut versions of current IV drugs, which I think in the UK is starting now with some IV.
Subcutaneous, right?
Yes, that's right. Okay. Yeah, particularly important at the moment, of course, with all the COVID-19 pandemic going on, that we really need a way perhaps of encouraging patients to be able to have their medications without coming into the hospital. So that's something that is quite exciting for patients. Again, there are two camps of patients that prefer to have the IV, patients that prefer the self-administration. But right at the moment where we are, I think there is certainly an appetite for patients, understandably, to be able to have their treatments away from the hospital. So
is that your experience as well, Max, that patients would rather avoid those visits to the hospital and there's a drive towards doing more things at home?
So definitely. So we have a lot of discussion always also with patients, for instance, when they are starting to get treatment for anti-TNF or with anti-TNF antibodies, where you maybe can choose between some therapeutics that are intravenously, like infliximab, or some that are subcutaneously. And there's certainly a broad range of patients that prefer the subcutaneous administration to be able to do it at home. That's definitely the case. And I think it also applies to other aspects, like also maybe diagnostic or activity evaluation and things like this. Um, yeah, it depends on how the future will be here. So at the moment, I would say it's definitely good also to have regular patient visits, or clinical visits for the patients. But I mean, the same way as we talk here together today via the Zoom conference, this might change also in the clinical situation or context in future times too. Let's see.
On that note, we have a question that's come in. It's not particularly related to treatment, but we were talking about non-invasive monitoring, and you mentioned the optoacoustic imaging. Someone is asking, is optoacoustic imaging appropriate for use with young people who have IBD? Will it be something that's available to all age groups?
So definitely, I mean, we need to kind of state here also that it's really something we're evaluating at the moment. So we made a very large clinical trial here in Erlangen where we tested the technique in over 100 patients, and it worked quite well. It's a very short procedure. It just takes a few minutes. And we had another trial now that was actually done by the pediatrics department on testing patients with muscle dystrophy or dystrophic diseases here. And it worked really well in pediatric patients here in this situation too. So we think that it also could work quite well in pediatric IBD patients. Especially since it's non-invasive and the procedure alone does not contain much risk. So you use laser light for the excitation. This is a high energy laser light, but we have no evidence also from kind of our partner, iTheraMedical, who is kind of developing the system, that this is somehow harmful for the skin. So you should wear an eye protection, so like googles, for instance, during the procedure, but that's actually everything. Besides that, it's a really very uncomplicated and easy to perform procedure.
What information are the doctors looking out for when they scan IBD patients? So you mentioned that the blood count correlates to inflammation, and what information does that give about the progression of the disease?
So basically, it's a kind of a mixture of ultrasound and this optoacoustic information or optoacoustic imaging. So with ultrasound, we actually kind of evaluate individual parts of the intestine, and we want to see if they are thickened. So this is something we kind of see very regularly in IBD patients, no matter if it's Crohn's disease or if it's ulcerative colitis. And then optoacoustic imaging tells us how much hemoglobin is kind of accumulating in the intestinal wall, and this is something we then kind of quantify. So we kind of put a small box on the intestine, and then we get from the device a value that tells us this is kind of a higher or lower signal. And this is then kind of the value that we use to either compare the patient kind of response to the treatment before or afterwards, or that we can use also by comparing different patients to see, is this a patient that has rather higher activity, lower activity, or even remission. Regarding therapy, this is something we just are testing at the moment. The kind of situation at the moment where we just want to know, is it high or low activity or remission? This is something we have previously done and where we know it works quite well. And it shows really good correlation also with kind of endoscopic disease activity or histology.
Okay, thank you. Azucena, I wanted to go back to your the points you were mentioning earlier about the underlying knowledge of the disease and how the mechanisms of disease are different in different patients. I'm wondering, how detailed is the general knowledge that we have about the disease and its biological mechanisms, and how must this knowledge improve so that we know how to design more specific, more safe, and more convenient treatments?
Well,
So the question,
that's a big question. I mean, we certainly have a lot of information, but the problem. I think the challenge with this disease is the tissue that it affects. It's a very sophisticated tissue. So the intestine has many functions at the same time, and so this when we try to understand what's going on in an intestine, it really takes a lot of. I think it's like a big puzzle, and we just have a few pieces. Our resolution of the information that we have even today is not enough, I think. So it's a very complex tissue that has cells that are devoted to mostly digestion and taking in nutrients, but also has a lot of cells from the immune system, which is a very complicated system. It has a lot of nervous, cells that transmit signals, to our central nervous system. So that's why, a lot of psychological issues have a lot to do with how we feel intestinally. So there's many different layers to this. And now, even understanding the healthy gut, it's very complicated. If you take, the intestine in health, it has all these cells. Now, when you add to that the inflammatory response, which in each patient, like what you mentioned, it can be different, or it can be driven by different signals, then you have a picture that is tremendously complex. And so when we try to do science, we start working with cells coming from a patient or a small biopsy. So we're just really taking a snapshot at that moment in that patient and trying to, get the full picture of the disease from that. So even though we use a lot of, we try to do this through, working through many different techniques, working with different people, collaborating like in all these European projects, it's still a daunting task. I think that I'm not, I don't want to sound pessimistic at all. I mean, I think that we know a lot more than we did 20 years ago, for sure. And that, as everything else, I think it's going on. Science is progressing exponentially. So the techniques that we have today to study tissues, even when I look at my, at my own career, when I started what I was doing to what I can do now because of what it's available, it's really, really impressive. And so I think that we will know more, in the next 10, 20 years for sure, obviously. But I think it will not just be a linear progression. I think it's going to be exponential. But, the challenge is, I think the main challenge is what I try to say is that we're studying a very complex that has a lot of inputs. And, we say this, and I don't know, I know that many people maybe are not familiar, but in when you do research in diseases, many of us use animal models, so experimental models. And so if you look at all the models that are available for colitis, for example, there's many. And that's because anything that you can disrupt, there's so many things you can disrupt that end up with an intestinal inflammation in our body. And so I think that's why patients are so heterogeneous, because the reason why they develop
it's a chain of molecular events, and the failure could be at any point in that chain. It doesn't have to be always at the same one. Sorry, go on.
Yeah. So we don't have just one cell or one cell doing one thing, and even the same cell may be doing completely different things during different times. And we have about 30,000 genes in our genome that are being expressed, and not all of them, obviously, but at least 1,000 or 2,000 of those can be involved in the processes that take on in our gut. And so what we find with models, and you can see it as I'm just saying this, so it's an example of how this is affected, is just that when you have models of mice that lack one gene or have a mutation in these or whatever it is, and sometimes it is very diverse things, they can end up developing colitis, what we call colitis in mice, which is intestinal inflammation. And so to me, that's a sign, or it's a clue that we are really dealing with something that the intestinal gut, the intestinal equilibrium, what we call homeostasis, to maintain that, it requires a huge amount of effort from our body. And it does tremendously well. Most of us can deal with life, with events, with changes in our diet, with stresses, and with things, and will not develop this disease, will be healthy, will be tolerant to everything, will not mount an immune response. But you can break this equilibrium doing many different things and all end up at the clinic, diagnosed with Crohn's disease.
Yeah. So understanding these processes is, as you said, very complicated because of the heterogeneity. And I guess it's also quite difficult for patients to realize the importance of basic research because it seems so far away from what they are living day-to-day life. So the things that you are doing in the lab, simply to understand how the gut works. You were saying even the healthy gut is difficult to understand, never mind the inflamed gut. So getting this basic research knowledge down the line and knowing that it will impact later must be quite hard. And Rachel, perhaps from your point of view, I'd like to know how do patients stay on top of, obviously patients aren't scientists, they don't have to know about the biology, but I imagine a lot of them do end up knowing a lot about how their body works and how their inflammation works. They sort of become lay experts in immunology. Where do you get that information from? How do you think there's a need or desire to understand more?
Well, I think, particularly again, when you've lived with a disease for a long time, you do become, a self-appointed expert simply by dint of the fact that, as you say, it's happening to you, it's your body, and therefore certain things that are going to work and aren't going to work. So there is, I think you get two types of people. You definitely get people that buy into, want to know all the information, and then you get other people, of course, that don't, and they just want to, they're happy to go and have their treatment and be told what to do, and that's fine. But other people want to play an active part in it, and I don't think it's necessarily a right or a wrong there. I think it has to be what really chimes with an individual's needs in that way. But in terms of getting patients interested in research and the basic science of it, I think that we need, as I was saying earlier, a conduit from the clinical side into the patient community, because you can't really expect patients to understand the more technical side of things. So how do you do that? Well, you can have patient champions, advocates. You can have
such as yourself,
basic educational sessions. Well, it's very kind, thank you. Basic educational sessions. You can have patient information leaflets, but however you do it, it has to be in a language that the patients are going to both understand and be inspired by, because if you can't inspire a patient, then you're not really going to get them to engage with their disease. So however you do it, the important thing to my mind is the language that you use, and having somebody being able to translate that into a way that patients want to come on board with.
Right. Well, we have about 5 minutes left, so I think to wrap up, it would be good then if our experts can tell us in a language that patients will resonate with, it, something that will inspire them, as Rachel said, to realize the importance. Perhaps Max, you could tell us how you think that these new techniques in monitoring and in diagnostics will improve patients' prospects, both in terms of how the disease is going to evolve, but also just their life quality.
So in kind of relating to our project, which we are kind of somehow explaining here in this meeting, Euphoriah, the main aim would really be to have the possibility to do monitoring of treatment and the course of disease with kind of a lower requirement for performing endoscopy. So this would be something we would hope for. We don't want to kind of substitute for endoscopy, and I don't think that we can do that rather quickly, but it would be already an advantage if you maybe can perform something else instead of every second or 2 out of 3 endoscopies. That would be an advantage. So what we would hope is maybe that we can perform, or that we know if we perform already such a technology like what I explained here today rather quickly, for instance, after starting a new treatment, and then with kind of a very objective method telling patients shortly after starting the therapy if the therapy might work or not by predicting it, but by maybe seeing a response earlier than maybe clinical signs might show it, or even other imaging techniques or blood values or so can do it. So this would be a hope that we have. If it turns out to be true, or if we can fulfill this, the future will tell.
Okay, right. And how about you, Azucena? How would you say that basic research is going to benefit patients' lives, and how can that message be put across?
Well, of course, I'm an advocate of basic research. So I think, as I tried to say earlier, I think there's no, there's no other way around it. We cannot even think about developing any new technologies like what Max is explaining, or any new drugs without this basic research. What we have learned is that we need to know more about the drugs that we already have, not just necessarily get new drugs, but also about the new drugs that are coming. And I think that we have learned a lot. The positive thing is that there's been such an effort, and I think it's not just human effort, but economic effort. I think there's a lot of now funding going into IBD, and you can see our consortia, for example, and many others from the European Union. There's definitely an interest in this disease that's raised a lot in the last few years, and that means that the more resources we have, we can definitely apply the better science to this, which is, in my opinion, absolutely necessary.
Excellent. Well, thank you very much, everyone. Thank you, Rachel Sawyer from the UK. Thank you, Dr. Azucena Salas on behalf of New Deal, and thank you, Dr. Max Waldner on behalf of Euphoriah. It was very nice to speak to you today. Fingers crossed for the future of IBD.
Okay, thank you.
Thanks, Bruno.
Thank you.
Have a good afternoon. Goodbye.
You too.
Bye.
Thank you. Bye.